So, good afternoon, guys. We'll start the lecture. So, I am Rishabh, and today we'll start with the week 4 of the course Host Pathogen Interaction by Professor Himanshu Kumar from IISER Bhopal. Okay, so in this lecture, we'll be mostly discussing about the main focus will be about cytokines. Okay, so I hope my voice is audible and my screen is also visible to you guys. Okay, so what are basically cytokines? Okay, so cytokines are a group of proteins, so they lie in protein fraction of the biomolecules in our body, which facilitate cell to cell interaction. That means immune cells in particular. Okay, communication or interaction for development and immune responses. Okay, so these molecules are basically signaling molecules that helps in signaling between different cells, or that that can be because of any pathogen or any immune response. Okay, so this is the main function of what is a cytokine. Now, these cytokines, how these actually uh, like im- uh, import their functions, they can be autocrine, paracrine, or endocrine in nature. Okay, so what are these? Endocrine means whenever any protein molecule is released into the blood, it reaches to a distant cell. Okay, and it facilitates its function. So these cytokines can be endocrine, that means they can be released in blood in under circulation, and then they can reach to a distant cell and they impart their function. They can be paracrine, that means the cell that is secreting them will allow it to function on a nearby cell also. So that is called as paracrine, and this is short lived. Okay, so in terms of paracrine, it means that once the cytokine is released, it will directly act on the nearby cell and its function will be gone. Okay, now the last one is autocrine. That means the cells that are secreting the cytokines can directly have that function on them itself. Okay, they have the receptor, and the cytokines can directly act on the cell that is secreting. That means autocrine. Okay, so why did this happen? So that whenever there is any immune response that need to be taken care of, then in that case the cytokines help the cells to increase its production. Okay, that's why this autocrine function is very important. Then mostly all the cytokines are autocrine and paracrine in nature. Apart from this, IL-2 can actually function in all the three conditions. It can be endocrine, it can be paracrine, and it can be autocrine also. Okay, so here are some of the properties of the cytokines. For example, these are low molecular weight protein or glycoproteins. Okay, so most of the immune system related molecules or receptors, these are glycoprotein in nature. Okay, why is a glycoprotein important here? So that it will be actually like resistant to the attack from proteases. That's why most of the immune system related proteins are glycoproteins. Okay, these are secreted by WBCs mostly, and in response to any intrinsic or extrinsic stimuli. Then these cytokines are short lived. Okay, they just function and then they go away. Then the cytokines regulate the intensity and the duration of the immune response. Okay, so whenever there is any intense cytokine storm that happens, that means there is a huge stimulus for their production. Okay, so they regulate the intensity as well as the duration of the immune response, and they show their effect by many means. Okay, that we will see in the next slides. So the here are some of the functions how a cytokine actually works. So first is pleiotropy. That means one cytokine will have multiple functions. So multiple functions or multiple targets. Okay, so this is the first thing how a cytokine actually acts. So here is the example of IL-4. So for example, it has a B cell that leads to its activation, proliferation, and differentiation. Second target is thymocyte. That means it leads to its proliferation. And the third one is mast cell that also IL-4 also helps in its proliferation. So this is a pleiotropic effect of IL-4 on B cell, thymocyte, and mast cell. And in all the three cases, it is helping in proliferation. So here the target is same, but the function is target is different, but the function is same. Okay, so this is the pleiotropic effect of the cytokines. Then we have the redundancy. Redundancy means many cytokines will have similar function. Okay, and this similar function is basically done in different conditions. Okay, for example, here we have IL-2, 4, and 5 that are acting on the B cell and they helps in its proliferation. Okay, so this is called as redundant function of the cytokines. But in nature, these three cytokines can be produced from different cells under different stimuli. Okay, so for example, IL-2 is only produced by Th1 cells. Okay, and it helps in differentiation of uh, it helps in differentiation of T cells also. IL-4 on the other hand is secreted from Th2. Okay, or from the mast cells, and IL-5 is secreted from Th2 and mast cells. So as you can see here, the source of the three cytokines is different, but their function is same, that they are helping in proliferation of B cells. And as this says that source is different, that means the stimulus is different. Okay, so this is the second case. The third case is synergy. Synergy means when two cytokines act simultaneously on a single cell, then it can help to actually function on a single thing. For example, here IL-4 and 5 acting on the B cell or the plasma cell, so that it can induce class switching to IgE. Okay, so that means when both of these IL-4 and 5 act on a plasma cell, then IgE will be produced. So this is called as synergy. Now we have another one, that is antagonism. So antagonism means there can be some cytokines that can function in opposite manner. For example, here IL-4 increases the IL-4 increases the proliferation and class switching to IgE. But here IFN gamma, that means interferon gamma, it inhibits the B cell for producing the for uh, class switching to IgE that has been induced by the IL-4. Okay, so they both are antagonist in nature. That means they have opposite function apart from these. Okay, then we have cascade induction. So here what we have is we have a Th cell, that means a helper T cell that produces interferon gamma. So this interferon gamma acts on a macrophage. So macrophage then produces IL-12. This IL-12 then again activates the Th cells to produce IFN gamma, TNF alpha, and IL-1, 2, and other cytokines. Okay, so this means that one cytokine is not enough for producing these particular cytokines for the immune response. There are other cytokines that are also involved in producing the other cytokines. Okay, so that is called as cascade induction. That is the fifth function. So this is how these cytokines actually act in our body during any immune response. Okay, so if you guys have any doubt, you can ask here. Okay, so let's move on. So now we'll discuss about some of the cytokines and what are their main functions in a immune response. So the main functions of the cytokines are development of cellular and humoral immune responses. That means they help in specific cell proliferation. Okay, then help the plasma cells to produce specific antibodies. And the third one will be complement activation. Okay, so these would be the three main roles of cytokines in cellular and humoral immune responses. Then they can induce inflammatory response. Yes, the inflammatory response is basically production of large amount of cytokines. Okay, then we have regulation of hematopoiesis. Yes, which type of cells to be produced. So these are actually done by the cytokines, and the type of progenitor cell will produce which type of cell is also taken care by the cytokines. Then control of cellular proliferation and differentiation of different type of immune cells. Okay, just for example, eosinophils survival and differentiation is done by IL-5. Okay, so this is how the different cytokines can help the differentiation of different cells. Okay, then healing of wounds. Okay, so this also is done by different types of cytokines. Now, there are two kinds of immunity in our body. First is the innate, and the second one is the adaptive. So there are different set of cytokines that play role in different types of immunities. Okay, so in the innate immunity, we have in cytokines like interleukin 1, TNF alpha, IL-12, IL-6, interferon alpha, and interferon beta. Okay, so this is the source of these particular cytokines that takes place that are produced from the body, and this is the function that they do. So, for example, IL-1 is helps in inflammation, fever, induction of acute phase proteins. That TNF alpha helps in inflammation, induction of acute phase proteins, cachexia, and neutrophil activation. IL-12, it induces or influences adaptive immunity. It is produced, it can like direct from the NK cells. Then IL-6 is also induces acute phase proteins, and then proliferation and antibody secretion of the B cell lineage. Okay, then we have the antiviral state IFN alpha increases MHC class 1 expression, and then IFN beta also have the similar function. Now, moving on to some of the cytokines of the adaptive immunity, we have IL-2 that is secreted from T cells, help in T cell proliferation. main function. Then IL-4, these are also, like I said, it is secreted from Th2 and mast cells, helps in Th2 differentiation, and main function is isotype switching to IgE class. IL-5 also secreted from Th2. It major function is eosinophil activation and generation. Then TGF beta, these are produced from T cells and macrophages, and they inhibit T cell proliferation and effector function. So they inhibit B cell proliferation, promotes isotype switching from to IgA. So IgA is produced basically in the M cells, okay, that are present in the malt. Okay, then we have IFN gamma. These are secreted from Th1 cells, CD8 positive cells, and NK cells, and they activate macrophages, increases expression of MHC class 1 and 2 for antigen presentation, and then increases the antigen presentation. Okay, so these are some of the cytokines that are present in the adaptive immunity, play an important role in adaptive immunity as well as in the innate immunity. Okay, so as you can see here, most of the immune response that takes place in our body. Okay, so as in immune response, saying is a simple term, but it has many subtypes. Okay, for example, different cell proliferation and its function, different antibody production, then production of the acute phase proteins. Okay, so likewise, different cells, different antibodies requires different cytokines. Okay, so that's why different cytokines are important for different cellular functions and different immune responses. Okay, so now moving on. uh, So here we start some of the questions related to what we have studied just now. Which of the following are not the function of the cytokines? So what can be the correct answer? Okay, so as we studied just now, so cytokines help in development of cellular and immune responses. Okay, then production of inflammatory response and healing of wounds, and control of cellular proliferation and differentiation. So all these three are the functions of the cytokines. So the question states which of the following are not the functions of the cytokine. So the answer would be none of the above. Okay. Sorry, it would be fourth. All of these are the functions of the cytokines. Okay. Moving on to the next question. Which of the following is a non-immune function of the cytokine such as IL-8 and MCP1? So what can be the correct answer? So both of these cytokines, IL-8 and MCP-1. So IL-8 is interleukin 8, and this is monocyte chemoattractant protein 1. Okay, so these two are mainly produced by the macrophages, and their major role include regulation of the ovulation, follicular development, and steroidogenesis. Okay, so as we have studied till now, that the immune function is the main function of the cytokines, but some of the cytokines are also involved in non-immune functions like regulation of the ovulation, follicular development, and steroidogenesis. So the correct answer is option number 1. Okay, so if you guys have any doubt, you can ask. Okay, so let's move on. Now we are moving on to the immune mediators. Okay, so there are some more molecules that are involved for the immune function to happen, that are the immune mediators. So we have the first class, that is vasoactive amines. So vasoactive means that they actually are involved in the vasculature, or they actually dilate the vessels. Okay, so that is the major meaning of the vasoactive. So there are histamine and serotonin. So this is the structure of the serotonin, and this is the structure of histamine. So these two molecules are basically vasoactive, and they can mediate the immune response in any conditions. Now, serotonin is present in hematopoietic stem cells and in high abundance in platelets, and it regulates inflammation and immunity by adding on serotonin receptor that are differentially expressed on the immune cells. Okay, so this is the function of the serotonin. Then we have, it is a potent chemoattractant that recruit immune cells to the sites of the inflammation. Okay, then it alters production and release of the cytokine as well as cell activation and proliferation. And the immune cells, including mast cells and T lymphocytes, have the capacity to synthesize and release serotonin. So these are the major functions of the serotonin. It helps in regulating inflammation and immunity, as well as it attracts different innate immune cells to increase the immune response. Okay, then we have studied for histamine. Histamine is a major molecular mediator in inflammation and allergy. Okay, so this is what happens when an allergen sits on any of the eosinophil or any of the allergic prone cells. Then they release vasoactive amines like histamine. Okay, now this histamine actually acts on smooth muscle cell, small blood vessels, mucous glands, blood platelets, sensory nerve endings, and eosinophils, so that they can actually dilate. Okay, so this is the major role of histamine in an inflammation on an allergic response. Okay, so now moving on to the second class, that is eicosanoids. So eicosanoids are basically lipid molecules. Okay, so we saw first the amines, histamine and serotonin. Now we have lipid molecules that are di- uh, uh, differentiated from different polyunsaturated lipids. like arachidonic acid. Okay, so in that case, these are lipid molecules that have a short-lived immune function. Okay, and they are involved in reproductive function, in the inflammation, fever, pain associated with injury or disease, formation of the blood clots, regulation of blood pressure, and gastric acid secretion. Okay, so these are the major function of the eicosanoids. There are three different types of eicosanoids that are basically studied in immune response: thromboxanes, leukotrienes, and prostaglandins. Okay, so these are type of terpenes that are present in our body. Then we have these are the structure of these three that are derived from the arachidonic acid itself. Okay, so these are the forms of lipid that are helping in different immune responses. Okay, just a second. So we saw amines, we saw lipids. Now we are moving towards peptides. Okay, so we have a peptide that is called as bradykinin. Bradykinin is a 9 amino acid long peptide that helps in regulating blood pressure. It is a vasodilator. It dilates the blood vessels. It plays an important role in inflammatory reactions, and it elevates vascular permeability and the cause of vasodilation in some of the arteries and veins. Okay, the similar function that it acts as a vasodilator in any of the functions, and it has a role in the inflammatory reactions also. Okay, now we have some proteins that are part of the complement system, that is C3A, C5A. So these are the fragments that are released when the complement pathway is going on. So to during activation, the complement proteins are divided into some parts. For example, C3 is divided into C3A and C3B. So C3B is involved in the complement pathway itself. C3A has some other functions. For example, they act as a signaling molecules to activate some of the other proteins. And moving on to some of the cytokines such as IL-1 beta, IL-6, and TNF alpha. So these are also protein molecules that act as cytokines for signaling. Okay. Then we have type 1 interferons. These are interferon alpha and interferon beta. So interferon alpha has 13 members and interferon beta is a single molecule. Okay, then we have inducible genes. So IFN inducible genes are IP10 and RANTES, and type 3 interferons are IFN delta, and it consists of three members only. Okay, so these are some of the cytokines that are proteinaceous in nature. Okay, so the biochemically they are proteins in nature. Okay, so let's move on to the question session. Which of the following statements regarding serotonin is or are correct? So what can be the correct answer? Okay, any more answers? Okay, so the correct answer is option number two. So it acts as a potent chemoattractant. It recruits innate immune cells at the site of inflammation. Okay, so basically, the first option that is there, that is serotonin regulates the inflammation by acting on serotonin receptors that are differentially expressed on non-immune cells. Okay, but in case of inflammation, what happens? The major function is taken care of by the immune cells. Okay, so that's why the first option is incorrect. Then non-immune cells have high capacity to synthesize and release serotonin. No, this is also not correct. So the correct answer is option number two. Okay. So any doubts in this question? Okay. So let's move on. What is the primary impact of vasoactive amines like serotonin and histamine in the immune system? So what can be the correct answer? Okay. Any more answers? Okay, so the correct answer is option 4, as we have studied just now. The serotonin and histamine both are involved in regulating the inflammation and the allergic reactions. Okay, so this is the correct answer. Let's move on. What among the following is not a icosanoid? Okay, any more answers? So the options are prostaglandins, thromboxane, lipoxins. So these are the eicosanoids and leukotrienes. So these four are the eicosanoids. Thrombin is a protein that is mainly involved in clotting. Okay, so the correct answer is option number 4. Okay, so let's move on to the next question. Which of the following statements are correct? So what can be the correct answer? Okay, any more answers? Okay, so let's see the options. IL-2 exert its effect in an autocrine, paracrine, and endocrine manner. Yes, this is correct. So it can exert its effect in all the three cases. Okay, MCP1, that is monocyte chemotactic protein 1, are involved in follicular development, atresia, ovulation, steroidogenesis, and corpus luteum function. So yes, this is also correct. The adrenal cortex, adrenocortical chromaffin cells produce cytokines such as IL-1, 6, TNF alpha, leukemia inhibitory factor, and IL-18. Yes, this is also correct. These two are the main examples that the cytokines can do non-immune functions also. IL-8 recruits neutrophils at the site of infection. Yes, this is correct. IL-1 beta promotes differentiation of naive T cells into Th1 cells. No, this is false. Okay, so IL beta have major functions in inflammation. Okay, IL-5 induces acute phase protein secretion. No, this is also not correct. So IL-5 is basically involved in eosinophil differentiation. Okay, so option one, 2, 3, and 4 are correct. So the correct options are option number 1. Okay, so any doubts in this question? Okay, so let's move on to the next thing, that is the inflammation that is associated with the cytokines. Okay, so basically what happens is whenever there is infection, then it increases the cellular and molecular factors releases the molecular factors from the traumatized or stressed cells or the tissues. Okay, so these factors that are released, then that leads to inflammation. Okay, so this inflammation can be related to the innate immunity of the cells. Okay, and the second thing is the then there is outcomes. Okay, so this is how an inflammation happens. Okay, then we have the cause of the inflammation. So cause of the inflammation can be any microbial infection, unfavorable living and non-living entities challenge, tissue injuries, or loss of homeostasis by intrinsic and extrinsic factors. Okay, then we have impact on physiology. So microbial infection can lead to pathogen clearance. Okay, so what will be the pathological outcome of that? It will lead to sepsis, tissue damage, and some of the autoimmune diseases. Okay, unfavorable living and non-living entities, these are helping in clearance of living and non-living entities, so they help in the body helps in clearing of those entities, and these lead to allergy and anaphylaxis. Okay, so anaphylaxis is basically a immune response that is most intense, right? Then we have tissue injuries. So tissue injuries lead to tissue repair. So body tries to repair the tissue, and this leads to tissue fibrosis, which may result in cancer. Okay. Then we have sorry, loss of homeostasis by intrinsic and extrinsic factor. So this leads to adaptation to stress or restoration of the homeostasis, and homeostasis associated diseases, as example heart failure and diabetes. Okay, so these are the major causes of the inflammation and what is their pathological outcome in an inflammation. Okay, so now moving on to the different cytokines and there are two types of inflammation. Okay, so one is acute and the other one is chronic. So one is acute and the other one is chronic. Okay, so acute inflammation is due to PAMS or DAMs, that is pathogen associated molecular patterns, or disease associated molecular patterns, that can be cellular stress or trauma. Okay, and DAMs are basically the systemic chronic inflammation is due to DAMs, that means exposomes, metabolic dysfunction, or the tissue damage. Okay, so acute inflammation means immediate. Okay, and chronic means long term. So this is the trigger for the acute inflammation and this is the trigger for the chronic inflammation. So any disease condition will lead to chronic inflammation. We have a duration, it is short term, it is persistent or non-resolving. That means it leads to long-term effects. The magnitude of the acute inflammation is high grade and it is low grade. Then outcome is healing, trigger removal, and the tissue repair. And in this case, it leads to damage of different organs. Okay. Then the acute inflammation is not related to any age, but chronic inflammation is. So, for example, if there is any aging related issue, then chronic inflammation takes place. Okay. The biomarkers of acute inflammation are 1L6, IL6, TNF alpha, IL1 beta, and CRP. Okay, and the for chronic inflammation, no standard biomarkers are there because in this case there are many that they are disease associated. Okay, so for example, in diabetes there would be some different biomarkers, and in case of cancer it would be some different biomarkers. So that's why in the chronic inflammation the biomarkers are not specifically defined. Okay, then in case of inflammation, what happens when it turns from towards chronic inflammation? So these are the major factors that takes place. So a breakdown of immune tolerance, a result of major alteration in all tissues and organs, result to major alteration normal cellular physiology. So these are the major hallmarks of the chronic inflammation. Then it is a result of impairment of, just give me a minute. It is result of impairment of normal immune function leading to increased susceptibility to infections and tumors, and a poor response to vaccines, increase the risk of various non-communicable diseases in both young and older individuals. Then during pregnancy, if the chronic inflammation takes place, the child then the child that is born actually has some serious developmental consequences, increasing the risk of non-communicable diseases over the lifespan. Okay, so that means during pregnancy, if there is any type of inflammation that can lead to any serious consequences to the child, so that can be avoided. So this is the major hallmarks which takes place when a body shifts towards the chronic inflammation. Okay, so if you guys have any doubt, you can ask. Okay, so let's move on. Which of the following conditions is most likely to result from dysregulated cytokine production? So, what can be the correct answer? Okay, any more answers? Okay, so yes, the correct answer is autoimmune disease disorders. In that case, what happens? There is a cytokine storm that takes place. Okay, so for example, this is a case with the multiple sclerosis. Okay, so what actually happens here in the brain? There are multiple cells that enters. Okay, so these are the macrophages that enter into the brain tissue, and it leads to the production of different types of cytokines that go and directly target the neurons. Okay, so this is known as a cytokine storm, and this mostly takes place in autoimmune diseases. Okay, but whenever we have a viral infection also, then in that case also these things can take place. Okay, so this is how the dysregulated cytokine production happens in an autoimmune disease. Okay, so any doubts in this question? Okay, so let's move on to the next question. How has genetic engineering impacted cytokine therapy? So, what can be the correct answer? Okay, any more answers? Okay, so let's see the options. it has decreased. So basically, genetic engineering means that we can produce different proteins outside. Okay, so this is the major use of the genetic engineering. So we can actually engineer proteins for their high yield or their activity. Okay, so genetic engineering has made feasible to produce essential proteins for therapeutic purposes, and it can be given as a injection to the body. Okay, so yes, the correct answer is option number 3. Okay, so any doubt in this particular question? Mm. Okay, so let's move on to the next question. What are some of the diverse applications of interferons in medical treatment? So, what can be the correct answers? Okay, any more answers? Mhm. Okay, so the correct answers are 1, 2, 4, 7, and 8. Okay, so interferons are basically used to treat viral infections in cancer therapy, immunotherapy, and neurobiological conditions like multiple sclerosis and reducing neuroinflammation. Okay, so these are the major applications of interferons that we see. Then it is basically seen in virus viral infections, for example in hepatitis B and C. Okay, hepatitis B and C are caused by the viruses along with the ribavirin, so it can be administered along with ribavirin. Then in case of hairy cell leukemia, A type or A type of B cell leukemia, CML, and Kaposi sarcoma. So we can actually give the interferon therapy to interferons to these particular conditions. Then in multiple sclerosis, okay, follicular non-Hodgkin lymphoma, skin T cell lymphoma, multiple myeloma, hereditary in immunodeficiency, chronic granulomatous disease, okay, in which there is NADPH deficiency, NADPH oxidase deficiency. Okay, then we have osteopetrosis. Okay, so all these conditions, the interferons can be given as a therapy. Okay, so any doubts in this particular question? Okay, so let's move on to the next question. Which of the following are considered pro-inflammatory cytokines? So what can be the correct answers? Okay, any more answers? Okay, so the correct answer is option number 1, 3, 4, 5, and 7. Yes. So IL-1 alpha is help in it initiates perpetuating inflammatory responses, induces fever and vasoconstriction. IL-1 beta, the same in response in but vasodilation is here. Okay, so it leads to vasodilation. IL-6, it induces acute phase protein secretion, T cell and B cell growth maturation. IL-8, it recruits neutrophils to the site of infection. IL-12, it promotes differentiation of naive T cells into Th1, blocks angiogenesis, and activates cytotoxic T cells. Okay, so these are the function of different interleukins that are present here. Then we have TNF alpha, tumor necrosis factor alpha, that promotes leukocyte extravasation, induces fever, and promotes vasodilation. INF, IFN gamma, it activates cytotoxic T cells and macrophages, upregulates MHC2 expression in macrophages. Then MCP1, it is involved in monocyte trafficking, reduces monocytes and macrophages and NK cells. Then we have GM-CSF, granulocyte macrophage colony stimulating factor. It stimulates monocytic cells, promotes their survival and differentiation. So these are the common pro-inflammatory cytokines that come into picture during inflammation. Okay, so any doubts in this particular question? Okay, so let's move on to the next question. Which of the following is not the function of TNF alpha? So, what can be the correct answer? Okay, any more answers? Okay, so yes, the correct answer is option 3. Just now we studied that TNF alpha promotes in leukocyte extravasation, okay, from the blood vessels. Then it promotes vasodilation. It suppresses fever. It increases fever. Okay, so this is not the function of TNF alpha. So the correct answer here is option 3. So let's move on to the next question. What is the role of GM-CSF in immune cell development? So what can be the correct answer? Okay, any more answers? So the correct answer is option 2. It causes hematopoietic stem cells to differentiate into dendritic cells. Yes, this is the correct answer. Okay, so let's move on. What is the primary application of GCF therapy? Granulocyte colony stimulating factor therapy. Okay, any more answers? Okay, so the correct answer is 3. So GCF basically helps in neutrophil production. Okay, so whenever we have a disease, then in that case, GCF is given so that it can increase the neutrophil production. Okay, so let's move on to the next question. Which of the following best describes the function of type 1 interferons? So, what can be the correct answer? Hmm. Okay, so the correct answer is option 2. So, IFNs or interferons are basically important that they interfere with the viral replication and exhibit anti-proliferative activity. Okay, so this is the example where interferon therapy is also given. Okay, so let's move on to the next question. Which interferon is primarily used in the treatment of multiple sclerosis? So what can be the correct answer? Okay, any more answers? Okay, so the correct answer is option 2, that is interferon beta 1A is involved in the treatment of multiple sclerosis. Okay, so here are some of the examples of the interferons that are eh, given during different treatments. Okay, so their name, what is their nature, and what is their clinical applications. So we have roferon, that is interferon alpha 2A, that is involved in hepatitis B, hairy cell leukemia, Kaposi sarcoma, and hepatitis C. Intron A, that is interferon alpha 2B, so it is involved in melanoma. Then we have betaseron, that is interferon beta 1B, involved in multiple sclerosis. Evonex, that is also interferon beta 1A, that is also involved in multiple sclerosis. So these two are similar interferon beta only, interferon beta 1A, that is given to patients with multiple sclerosis. Then we have Actimune, that is interferon gamma 1B, that is given to GCD and osteopetrosis. Okay, so these are different interferons that are given under different clinical conditions. Okay, so any doubts in this particular question? Okay, so let's move on to the next question. Shifts in the inflammatory response from short to long lived, that is chronic, can cause which among the following situation? So, what can be the correct answer? Okay, so as we studied before, whenever an inflammation changes from acute to chronic, okay, so in that case, what happens is the body will actually tend to, that means there is impairment in immune function, and there is increased susceptibility to infections. Okay, so both these options are correct. So that means 1 and 3 are correct. Okay, followed by this, that uh, it changes the cellular physiology. Okay, the cytokine pattern, all these things are changed when the inflammation changes from acute to chronic. Okay, so any doubts in this particular question? Okay, so let's move on to the next question. Which of the following pairs is wrongly matched? So what can be the correct answer? Okay, any more answers? Okay, so the correct answer is option 2. That means basically iximab, that is Simulect, and daclizumab, that is Zinapex. It is not monoclonal antibodies against TNF alpha. Okay, so here are the agents that actually have a clinical application. For example, we have the agent name, then the nature of the agent. So this is Embrail, that is chimeric TNF receptor or IgG constant region, that is used for rheumatoid arthritis. Then we have Remicade or Humira, that is a monoclonal antibodies against TNF alpha, so that is also used for curing rheumatoid arthritis and Crohn's disease. Okay, so both of these are autoimmune diseases, and in that case, what happens? TNF alpha is basically secreted more because it's a inflammatory disease. Okay, so if we are targeting receptor of the TNF alpha to decrease the impact of that particular disease. Then we have Neupogen, that is GCSF, hematopoietic cytokine, so that it can stimulate the production of neutrophils, reduce infection in cancer patient as well as treated with chemotherapy and AIDS patients. Okay. Then we have Leukine, that is GMCSF, hematopoietic cytokine. It stimulates production of myeloid cells after bone marrow transplant. Then we have Numega or Neulasta, that is interleukin 11, a hematopoietic cytokine which stimulates production of the platelets. Then we have Epogene, erythropoietin. Okay, so this stimulates RBC production. So what happens is, in case of a patient that receive chemotherapy, that leads to the RBC lysis. Okay, and the production of the RBC is also reduced. So in that case, we give erythropoietin so that the RBC production is increased. Then we have Ankinra, that is recombinant IL-1RA, that is also used for rheumatoid arthritis. Then we have taclizumab or Zinapax, that is humanized monoclonal antibodies against 1L IL-1R, sorry 2R, that prevents rejection after transplant. Okay, so after transplantation of any organ, there is a term called as immune tolerance. Okay, so what happens whenever we transplant any of the organs from different individuals? So in that case, our immune system cannot recognize or cannot accept that organ. So for that, we give different treatments so that there will be the immune acceptance of that particular organ. Okay. Similarly, the second one, that is basiliximab. So it is a human or mouse chimeric monoclonal antibodies against IL-2R. So it also prevents transplant rejection. Okay. So these are some of the agents that are helpful in terms of blocking or providing the cytokines in different diseases. Okay. So if you guys have any doubt, you can ask. Okay, if no doubt, then this was the last question for today. If you guys have any doubt in the presentation or from the last week, you can ask. Okay, if no doubts, then we'll close the session for today. Thank you guys for joining in. We'll meet in the next class.